Sujan did a great job overviewing what AIM-High's mission is, so I'll just say that really the crux of what we're doing is what you're going to hear from this panel today, highlighting companies that are really at the interface of innovative cancer research and the industry that's charged with bringing forward new therapeutics, preventives, diagnostics for helping cancer patients. AIM-High is something that I got interested in personally because I'm interested in alternative business models and funding models to help catalyze that process. We did this at Scripps Research Institute where I was for 10 years. And another story for another day, but I'd love to talk to folks. We've just moved a few of us here to Baltimore to work on a similar model in partnership with Johns Hopkins and others called Blackbird Labs. So with that, I'm going to introduce our first speaker, Dr. Ignacio Acial. He's coming to us from Singapore, where he's the CEO of .bio.
Thank you so much. I'm gonna thank you. Thank you. Thank you all for being here. It's a great pleasure to present to you .bio. So let me get back a bit. Yeah. So this is .bio. So it's a company that we launched in 2018, and we focus on multifunctional antibodies and intracellular antibodies. So obviously these are very tough modalities. And we do this with a modular design approach, which is very unique to us. I'll walk you through what that means. So through that time, we've been able to put together a quite knowledgeable team of scientists first. We came from an academic endeavor first. And then over time, we brought in a number of biotech executives into our board. I'm also very glad that Sujan Bhatt has joined us also as a strategic advisor, and we get great help from that front as well. Yeah, so the company was spun out from the Karolinska Institute in Sweden and also from Nanyang Technological University in Singapore. It was part of my PhD work, actually, that we then eventually put into commercialization efforts. We have reached clinical stage with the company mainly through an out-licensing deal that we did of one of our molecules that was approved for clinical trials last month, actually. And it's a platform company. We generate a lot of assets, and some of those assets we develop internally, and some we partner. And through those partnerships, we already generated $200 million worth of deals to date. We just closed our pre-Series A round, and that's going to allow us to start pushing some of the internal molecules through efficacy studies in animals and also cell line development. So yeah, just to summarize what we do and what we don't do. So on the left side, if I can do this. Yeah, sorry, here. On the left side, you have the traditional antibodies. So the monotherapies, they target one thing. We all know what that is. We are focusing on the more complex antibodies that target perhaps two targets in the cancer space or 3 targets to try to avoid resistance and block resistance early on. And we're also looking at how we can make ADCs better by making them more specific with dual specificity. And finally, some area that's quite unique to us, which is intracellular antibodies. So as you guys know, so far, any intracellular target is the realm of small molecules. We're aiming to bring antibodies into that space. So I want to start with these numbers here. You probably know them. It takes about $2 billion to get one successful therapy through all the clinical trials and get an approval. But if we break that down, that's a very huge number, right? If we break it down in terms of how much it costs to bring any molecule through clinical trials, it's a much smaller number. We're looking at only 100 million if you just do the math of how much one molecule costs. So where is the difference? The difference is mainly on the failure rate we have in general to bring therapies. So because we fail so much, 85% of the time, we end up generating so much cost and obviously delays on getting new therapies to patients that need them. So if we look more specifically now at the oncology space and within that space at bispecifics, where we have more data, you can see the historical trends here on the left side of the number of trials that involve bispecific antibody. And that's growing exponentially over the years. We are now at more than 100 trials per year. So it's a very huge number for this space that is a relatively new space. But if we look at the number of approvals, it's still very limited. You can see We have a first approval in 2014, one molecule, then nothing for many years, and now a handful of approvals. So still, the success rate is even worse than the rest of the industry. We are looking at more than 95% of molecules that fail in clinical development. And this is still skipping all the preclinical work where we have even more failure. So the vision behind .bio is to be able to predict the molecules that will be successful in the clinic, so the multifunctional antibodies that will be successful in the clinic, by generating a lot of preclinical data that we can rely on. So let me unpack this a bit more. So if we think of how we develop these complex therapies right now, we typically gather a team of experts that think very carefully about biology and then decide on 2 or 3 targets to explore at one go and then combine them. And after that, we push those molecules through all the preclinical work and clinical work to realize at the end that they typically fail. So what we're proposing here is a different approach, which involves the ability to generate hundreds of bispecifics and trispecifics in parallel against different targets, still looking at the molecular biology and our understanding, but exploring a bit more broadly on what combinations we can make into those bispecifics. And after generating all those molecules, then have side-by-side testings of those molecules so we can make informed decisions of which one we should pursue into the clinic, and by doing so, then lower the risk of clinical failure. So this is the process that we have set up. It involves a lot more that I cannot go into details at this point. But it starts by generating a very large library of bispecific antibodies. So looking at a combination of 2 targets, let's call them target A, B, and then A plus C or C plus D, right? All the combinations we could think of. And then, of course, looking at the molecular biology to make sure that the orientation of these molecules also makes sense, right? Because there is a geometry that is required for these molecules. Then we have set up high-throughput phenotypic screens that involve different cancer cell types, different immune cell types, different donors, materials. And then after that screen, then basically we can decide which molecules have the most potency and then eliminate the rest and focus on those. And then convert them further into either trispecifics by adding a new set of specificities in addition to the previous 2 we found, or looking at then bispecific ADCs by looking then at different payloads we could use, the different drug-to-antibody ratios, and so on. Again, eliminate the majority of molecules that are weaker and focus then on the best ones to push into animals and then the best ones of those to go into human. So instead of focusing on just a handful of molecules that we try to push to the end as fast as possible, we start with a large repertoire early on that we test all side by side. We compare with FDA-approved drugs as well. And then we pick the very best through that process to bring into the clinic. And we believe this should reduce the failure risk in the clinic. This is an example of the kind of data we generate. So here we're looking at 58 bispecific antibodies compared with the monospecific building blocks. In this case, it's a colorectal cancer model. And we're looking at the level of cell killing that those molecules induce in the presence of immune cells. And you can clearly see that some bispecifics here on the left are much more potent than the ones here on the right. And the majority are better than the monospecific building blocks. And as a reminder, we designed all these molecules thinking that they should work. We looked at biology and thought these 2 targets should work together and have synergistic effects. But clearly, they are not made equal. So the idea behind the company is then let's focus on the best ones. I'm kind of taking some shortcuts here. Obviously, we do different models. We look at different cancer types. We look at different immune cells donors and so on. And then we decide on those that have consistently shown better results and then focus on those to then convert them into either trispecifics or bispecific ADCs. So again, to summarize, this is a very large market, obviously. It's a growing market. You've probably heard of bispecifics, trispecific, and antibody-drug conjugates. But there is very high failure rate, so there is clear need to improve our process here. And the idea here is to generate a large amount of preclinical data to validate which target combinations, which molecules should be pursued into the clinic. We have closed three partnerships to date. One of those, as I mentioned, has reached IND approval last month. And we are based in Singapore, so now what we're looking for is for partners that will allow us or help us come to the US and have an expansion here, where, of course, a lot of the biotech activity is happening. So if you are one of those that can help us bridge and come to the US, please approach me, and I hope we can make that happen. I won't have the time to talk about this, but we have made some big developments in the intracellular antibody space. I would just want to highlight our paper from last year in Nature Communications, highlighting then an antibody we developed for an intracellular target that is able to inhibit cancer cell growth in that space. Please look it up. Hopefully in the future, as we have more data, I'll start talking more and more about this as well. And with this, I would like to thank you all. This is a summary of our partners. Of course, we have AIM-High and the Asian Fund for Cancer Research that have helped us along the way since the beginning. So I thank you all for that effort, especially Su Juan. I want to mention also Ella, and then a number of the advisors that we have within this group. Thank you very much for your time.
Great, thanks, Ignacio. We're going to hold questions until the end. We have about 5 or 10 minutes at the end. So the next speaker is Dr. Stacy Blaine, who is the co-founder, CSO, and acting CEO of Concarlo Therapeutics.
Okay. All right. Well, thank you, Sujan, for inviting me here today. Very proud to represent Concarlo. Let's see what we got going here. Okay. So we are inhibiting the protein P27, and essentially we're doing this, to generate what we think it will be the first inhibitor of CDK2/4/6. And we're all in on this target P27. So our entire pipeline is really based around this target. So hopefully you can see all this, but what CDK4/6 and 2 sit at the bottom of what I call the oncogenic funnel. So all of the signals that drive a cell to proliferate, that run from the cell surface, the receptors, they run through the cytoplasm, interacting with the RAS and the RAFs, the PI3 kinase, they all feed down to this hub that sits at the bottom that is really the restriction point that tells a cell whether it should divide or alternatively not divide. And most of the targeted therapies that we have in the era of precision oncology that recognize either the receptors that sit at the cell surface or the small molecule kinase inhibitors that bind to the RAFs and the MECs and the proteins that sit in the cytoplasm, when they work, they block that signaling and they block the activity of CDK4/6 and CDK2. But I ask you to think about CDK4 and CDK6 as a hub, because that's really the way they think of, the way they work. And whatever we do to CDK4 is actually felt by CDK2, and conversely, whatever we do to CDK2 is felt by CDK4. And so in the era of precision oncology, we've created a lot of monoclonal antibodies to bind to those receptors, the small molecule kinase inhibitors, and they work well. But we now, it was mentioned earlier today, the resistance problem. In the US alone, there are roughly 600,000 patients annually that will die of drug-resistant cancers, cancers that have no longer responsive to our current standard of care. And a lot of time that's because while we might target something at the cell surface, there's just too much room for that cancer cell to acquire mutations downstream of the blockade that we put on the cancer cell. For example, we might use a drug like Tarceva to inhibit EGFR, but that same cell could acquire mutations in RAS or RAF or MEK and push that cell out of a remission state into the resistant state. So that has sort of renewed our interest as a field to drug at the bottom of the oncogenic funnel, because the same way it's easier to block the flow of water by pinching off the narrow bottom of the funnel, it's going to be easier to block that flow by hitting CDK4/6 and CDK2. But I'm arguing that you have to hit them together. And the best evidence of that comes from our use of drugs like Ibrance, Verzenio, or Kisqali, the CDK4 inhibitors, which came to market in 2017 in the metastatic breast cancer space. Those are great drugs. They extend the progression-free window for our patients, but we know that the overall survival of those patients really hasn't changed as dramatically as we wanted to do. And essentially, almost 100% of those metastatic breast cancer patients will become resistant with time. And so the CDK4/6 inhibitors inhibit one half of the seesaw. They only block CDK4 and 6, but with time, CDK2 turns on and compensates. Again, reinforcing, if you really want to stop this pathway, you have to hit CDK4/6 and CDK2. Now, people have been trying to do that for over 40 years, but the problem is the CDKs are part of a very large kinome. There's 20 different CDKs. They look like all other serine threonine kinases, and so most of our attempts to drug with specificity have hit lots of other off-target other targets. And so at Concarla, we said we're going to take a very different approach. We're going to stay away from the conserved CDKs, and we're going to target this other protein that is present in this hub, this protein P27. And we're going to get P27 to do its job. P27 is the natural master regulator of CDK4 and CDK6 and CDK2. Now, P27 was actually discovered by Nelly Polyak, Joan Massaguet, Jim Roberts, and Andy Koff. And Nelly, I actually joined Joan Massaguet's lab now 25 years ago, dating us, Nelly, the week she was leaving to go on to do her postdoc. And so I've continued to work on P27 for the last 25 years with the goal of creating therapies to get P27 to act as an inhibitor. Now, how can we do that? So think about P27 as this door. And sometimes doors are open and sometimes doors are shut, but they're always present. And that's what P27 does. And so when P27 gets modified, when the key is present to the door swings open and the CDK active sites are on. But when the key is missing, when P27 is not phosphorylated, the door slams shut. And in fact, P27 forms interactions into the active site of the CDKs, shutting down CDK2, 4, and 6. We achieve our specificity by targeting P27 because P27 does that for us. It really only binds to CDK2, 4, 6, and leaves all the other CDKs alone. So at Concarla, we are creating classes of P27 inhibitors to bind to P27, block the key, slam the door shut, and turn CDK2, 4, 6 off to deal with that resistant state. And we do this by a variety of different ways. We have both large molecule drugs, so we have peptides that are packaged in liposomes. This is our lead asset, which is actually, we just finished manufacturing of that complex API, and we're about 18 months from our, I wish 18 minutes, right? From our first clinical trial. But we also, because we believe in the value of this target, we have small molecules against the same target, and we're starting to think about developing therapeutic RNA as well against this target. And so just to show you an example of what we can achieve with P27 targeting. This is a palbociclib-resistant breast cancer mouse model. So think of a woman, she has metastatic breast cancer, she was treated with 1 or 2 prior rounds of Ibrance, she fails off, her tumor starts growing. That's the model that the mouse that we have modeled here. They have very large tumors, about 700 millimeters cubed. If you treat these animals with palbociclib, we're plotting tumor volumes in the top, the tumor you can see just keeps growing, fully resistant. But if you treat with Concarlo's drug IPY, the tumor stops growing, flattens out. And if you treat with a combination of IPY plus Ibrance, palbociclib, you get massive tumor regression almost instantly. We drive the tumor to 0. It stays down for well over 40 days, even though we stopped treating these animals. This is some rebound due to tumor we didn't eradicate because we stopped treating them. And this then translates into what we need for this patient class, increases in overall survival. The big problem with drugging CDK2, 4, 6 is the tox issue, right? They're involved in many different cells. And in fact, that is the major, toxicity is the major dose-limiting problem with Ibrance. Patients are only on drugs for 3 weeks because they have massive neutropenia. Neutrophils are cells that we need to have. We don't let patients walk around without them. But we don't see that when you target P27. And so if you look here at untreated animals, and we're measuring neutrophil levels in gray, as soon as you start treating with palbociclib, the neutrophils crash to the bottom. But in green, Concarlo's IPY, tons of neutrophils. And in the combination therapy, where we were getting that regression, plenty of neutrophils. So we think, we'll enter the market with a very differentiated product, one that is hitting CDK2, 4, 6, has lower toxicity, has a different mechanism of action. We're actually causing tumor cell killing, and we think that this will provide an advantage to our patients. We want to initially go after those Ibrance-resistant breast cancer patients, but we're also looking at ovarian, very clear data that that is driven by CDK2. And we have very good data in other tumor types like pancreatic and non-small cell lung cancer in those resistant states, that they are also driven by CDK2/4/6, and our inhibitors will be appropriate there. So we do intend to expand into those different classes. And so where are we today? As I said, we've just finished our manufacturing of our lead product, which is very exciting. And we're ready to move into our IND-enabling studies, secure the IND, and then actually run our first phase 1A trial to demonstrate both tolerability as well as look for markers of efficacy in these patient populations. And so we are the P27 company. We envision that we will push IPY to the clinic for use in the tumors that are very clearly driven by CDK2 and CDK4/6. But then we also want to go after these tumor types where the CDK4/6 inhibitors are not currently used, such as pancreatic or glioblastoma. And then really thinking about the expansion potential in terms of combination therapy, that since these drugs will work downstream of all of these other precision oncology drugs, how can we put those drugs together to really increase responses? And so I have been studying this protein, as I said, for 25 years, and at first as a tenured faculty member, and then just this month I have actually retired, and I am now the full-time CEO of Concarlo. And so while I am not young, this is a young company, I guess I could say. And so I just want to end here, which was I was in the women's AIM High competition. In one of the first years, I was one of the winners of the competition, so I've appreciated all the support. And put the picture up here of my all-women's research team. And so I think it's really important to recognize organizations like AIM High, because as we've alluded to, it is only 2% of all women-run companies get venture funding. So hopefully with the help of this organization, we can change that. And so if anyone is interested in learning more about the exciting work we do at Concarlo, please find me. I'll be around later today. Thank you.
Great, thank you, Dr. Blaine. Our next speaker is Dr. Jeremiah Johnson. He is the co-founder, chair of the SAB, and a board director at Window Therapeutics, which is based on research in his lab at MIT.
Thanks, Matt. Well, it's such a great honor to be at this event again and to learn so much. It's just such an incredible experience. I'm an organic chemist, so I'm a fish out of the water, but I just feel so honored, and thank you, Sujuan, for allowing me to be here. And this time, I'm very excited to tell you about Window Therapeutics. Window Therapeutics, I figure out how to use this advancer. Window Therapeutics takes its name from the concept of the therapeutic window, which, as all of you will know, is the idea in drug development known as the therapeutic window, which defines the difference between the dose of a drug that is required to achieve a therapeutic effect and the dose that induces undesirable toxicities. And I think it's safe to say that regardless of the modality or the drug that you're trying to develop or the disease context, everyone wants to develop drugs with wider therapeutic windows. And in fact, as was mentioned, a lot of drugs, the vast majority that move to the clinic fail. And in most of those cases, that can be linked back to an insufficiently wide therapeutic window. And at Window Therapeutics, we hope to change that paradigm. And before I tell you how we are trying to do that, I want to first remind you of two of the different ways that scientists have previously developed drugs with wider therapeutic windows. And you've heard so many beautiful examples of these today. In fact, you just heard of one, so I'm not going to spend a lot of time covering these. But the first area is known as molecularly targeted therapeutics, wherein chemists, organic chemists, make molecular modifications to small molecules to develop drugs that are more selective toward binding a specific biomolecule, often a protein, associated with cancer cell survival, but not healthy cell survival. All right? Now, while these have been very successful and have certainly led to many new and promising therapeutics that are much better than the conventional chemotherapies that came before, they still often suffer from narrow therapeutic windows because it's just so rare that a pathway in cancer is 100% existent in the cancer cell and nowhere else in the body. Now, another common way to develop drugs with broader therapeutic windows is to use antibody-drug conjugates, which you've also all heard many nice examples of today, at least in their use in different contexts. ADCs follow a general formula wherein highly potent payloads are conjugated onto monoclonal antibodies, and here scientists are leveraging nature's evolutionarily optimized targeting machine, the antibody, to find bind to its target cell, and bring these highly potent payloads into the cell. But one of the challenges with ADCs is that because of the fact that the number of drugs that can be conjugated is fairly limited, usually anywhere between 2 or up to 8, as shown in this cartoon, it necessitates the use of extremely potent compounds. And in fact, in all the ADC world today, you can count on maybe 2 hands the different classes of mechanisms of action that are used. They tend to be generally highly cytotoxic compounds, and we rely on targeting of specific cells to bring those drugs to those cells. But it turns out that even with ADCs, it's been shown that about 99% of injected dose doesn't actually reach the target cell, and thus there is always systemic exposure to these highly potent compounds, which leads to a narrower therapeutic window than could be envisioned. So at Window Therapeutics, we're imagining, well, why not combine the beneficial features of molecularly targeted therapeutics, wherein the molecular structure defines an improved therapeutic window, with those of ADCs, where a monoclonal antibody achieves an additional improvement in cell-specific targeting to make a new class of antibody-drug conjugates. And that is indeed what we have done. Window Therapeutics has developed what we call the Antibody Bottle Brush Analog Macromolecule Conjugate, ABC, very simple. This is a platform that brings molecularly targeted payloads to their target cells. And this, we think, can be a new paradigm for cancer therapies. But first I want to show you how we can do this. And I have to touch on the fact that this is really a chemistry problem, which again is what I like to think about. The reason we're limited to 2 to 8 drugs per antibody, again, is because that high potency requirement. Well, most molecularly targeted drugs aren't that potent. In fact, they tend to be a factor of 10 or 100 less potent than the drugs used in ADCs today. So on a per antibody basis, you would need 10 to 100 times more drugs per antibody to achieve a similar effect. And you can't just go and attach 10 times more drug to an antibody, because as you may imagine, that would disrupt antibody function. And indeed, the canonical paradigm is really, it used to be you can't go beyond 4. Now people say, well, you can't go beyond 8. And that is simply because when people try to attach more drugs, you start to erode the antibody function. Moreover, most molecularly targeted therapeutics and drugs like them tend to be rather hydrophobic, so they can cross cell membranes, but that hydrophobicity causes structures like this to also lose stability and aggregate and lead to other challenges. So over the last decade in my lab in the Department of Chemistry at MIT, we've been developing various synthetic platforms to produce rigid chain molecules where on each repeat unit of those molecules, we can covalently conjugate molecularly targeted payloads and other classes of drugs through linkers that are selectively cleaved in disease microenvironments. But there's a problem with this approach too, and this has been attempted before, but the same problem arises, and that is that when we attach, let's say, 50 or 100 of a hydrophobic drug onto a linear chain, even though we overcome the problem of having to attach to many points on the antibody, we still face the problem that this would be very hydrophobic, unlikely to be soluble in water, and not useful as a medicine. And that is where we developed the synthetic technology that allows us, such that for every repeat unit of this polymer backbone, we have these blue side chains that extend into solution, and these are hydrophilic, water-loving side chains. And they are there to balance the hydrophobicity of these linkers and payloads. And the result is a platform that allows for unprecedented payload and target and linker modularity, because all of the features of the linkers and payloads are now expressed in the backbone of this bottle brush-like molecule. In fact, at the nano scale, these are literally like very small bottle brushes. And this allows us to swap things at will. And over the years, we've used over 40 different payload classes, including more traditional chemotherapies, molecularly targeted therapeutics, even molecular glues and PROTACs. We've used immune stimulants and many other kinds of drugs. Okay, now with this kind of payload versatility, the question a company is faced with, what do you use as your lead program? What's your first drug? And we were really drawn to the class of molecularly targeted therapeutics known as proteasome inhibitors. Many of you have heard of these compounds. They've been around for quite some time, most famously used as frontline therapies for multiple myeloma. But it turns out that since the 90s, it's been well appreciated that proteasome inhibition is a mechanism of action that's potent in a wide range of human cancers. And in fact, one of the reasons why it hasn't been used more broadly is because of its narrow therapeutic window. And that is best highlighted by this fact. It turns out that red blood cells have rather high proteasome concentrations, which are the targets of proteasome inhibitors. And so, especially for the classic drugs like bortezomib, which many of you have heard of, or Velcade, after injection, about 9 out of 10 molecules go into red blood cells, bind, and don't come back out. And as a result of that, that severely limits their bioavailability, which limits efficacy, and also leads to off-site toxicity and an overall net reduction in the therapeutic window, right? Nevertheless, the idea that proteasome inhibition could be used in the context of an ADC is very exciting because it would bring a brand new mechanism of action to this field and a mechanism of action that's orthogonal to most of the cytotoxic payloads used in ADCs today. For example, resistances that arise to topoisomerase I inhibitors would be overcome by proteasome inhibition. Now, at Window, we've developed linker technology that allows for the first stable prodrugs, so covalently linked proteasome inhibitors along the backbone of our bottle brush macromolecules. And we've produced these with lengths of 60, meaning there are 60 drug molecules per chain, which we then can conjugate to an antibody of choice to target that antibody specifically to a cell to unleash proteasome inhibition as a mechanism of action. And just to show you, this is some blood pharmacokinetics data, just to show you that our constructs, even with a DAR of up to 60, have half-lives that are right on par with what we expect for traditional ADCs, half-lives of a couple of weeks. And here on a dashed line just shows you where bortezomib, classic proteasome inhibitor, would be after just 2 days. So these are, this drug, after going into red blood cells, is cleared very rapidly. And with this construct in hand, we're able to generate data, as shown on this slide, where first we developed, we conjugated under our construct trastuzumab, which targets HER2, which you've also heard about in the presentations this morning. And with our ABC construct, compared to a range of controls that I won't have time to go into a lot of detail on, we get 100% survival with complete tumor regression in these BT474 HER2-positive breast cancer cell lines in mice. Now, this is very exciting that we are able to do this, but one thing that we've thought from the beginning is that this ability to have a drug-to-antibody ratio at least 10 times greater than the state-of-the-art may have another benefit, and that is that for cells that don't have high antigen expression, which may exist in the heterogeneous tumor microenvironment, it could be beneficial to have a higher drug-to-antibody ratio such that for every antibody-binding event, we bring more payload into that cell, therefore inducing a greater therapeutic response. And so we've tested our constructs in a different cell line, the HCC70 cell line, another breast cancer cell line that has very low HER2 expression. And what we find is there's a nice dose response here in light blue, dark blue, and in green, and the highest dose, which is safely tolerated by these mice. And we see a very nice therapeutic effect, reduction in the tumor volume. And what's notable here is we compared directly to TDXD, which was actually mentioned in one of the talks this morning. This is one of the clinically leading HER2-targeted ADCs. And you can see that even at a dose of 10 times less, we perform similarly, but because of safety, we can go higher and clear these tumors. And then I want to show that this really is a drug-to-antibody ratio effect. We are able to synthesize ABCs with 10, 30, and 60 drugs per antibody, and even at the same drug dosing, so we normalize such that each animal gets the same amount of payload, the DAR 60 significantly outperforms the others, confirming our hypothesis or supporting our hypothesis that a higher DAR can be beneficial in these low antigen expressing cell lines. So I'm out of time. I'm just going to leave with the last picture of the slide. Many of the folks are here in the room today, and we have a wonderful team of scientists. And again, Sujuan, where are you? Sujuan, thank you so much for the opportunity to be here. Thank you all.
Now we'll welcome to the podium Dr. John Luck from Arbel Bio.
Thank you, Matt, for the brief intro, and also I'm very honored to be here, invited by Suzhang and AFCL to showcase our company. Okay, next. Okay, in front of so many distinguished scientists, I prefer to give a very low dose of scientific information in this talk. So I'd like to share my journey as an entrepreneur, starting the company in 2016. So Arbel is a spin-off company from University of Hong Kong, where I graduated, and then later I went back to be a faculty at the Department of Surgery for 12 years before I started the company. So what I learned from the university, from the academia, is we learned the problem from our clinical practice. No matter how meticulous we practice, we operate, we remove the tumors, the patient still relapse, right, from the tumors. So we find the problems from the clinical practice. And on the other hand, so we discover opportunities, how to treat the patients, and we discover the finding the opportunities from the clinical specimen. We interrogate the clinical specimen. In fact, almost 2 decades ago, I started a biobank at the Queen Mary Hospital, collecting surgical specimen, not only from a tumor, but also from the adjacent peritumor specimen. And then we start looking at the specimen at the molecular level, from gene expression profile, genotyping, whole genome sequences, and then proteomics. And then we understand the disease biology, and then we also study the biomarker for better prognosis and diagnosis. And very strikingly, we find that actually the tumor stromal, the peritumor, actually has a much higher prognostic value versus the tumor. Actually, when I look at the data, I don't believe that. That means the tumor stroma play more important role for the patient's survival. And then last but not least, I think it's very important to identify the target. We had there are so many undruggable targets. So we need to find a good target for us to develop new drugs, new medicine to treat the patients. So our company started in mid-2016. So we landed at the Hong Kong Science and Technology Park. There's an aerial view, and you see the golden goose egg, and our company actually behind the golden egg. So one day we will take off. It's like a rocket going to take off to the sky. So over the past 5 years, we become from a preclinical to a clinical stage company. We started the first patient dose in Australia in 2022. And then we already expanded the trial to 2 sites in Australia and 1 site in Hong Kong Queen Mary Hospital where I was a faculty. And now we plan to advance to the phase 2 next year in the US and also in China and Japan. We try to file the fast track designations for these drugs. So what the problem we find is there's a huge unmet medical need for GI cancers. As you can see, colorectal cancer remains the top, the cancer mortality. And because of the aging population, the incidence will continue to raise, right? Not only in the United States, but also all over the world, colorectal cancer remains the leading cause of GI cancer, and also second to the lung cancer. But however, when you combine all the GI cancer subtypes, actually the incidence and mortality is higher than lung cancers. So if we can identify a target or drug that can cure 10 or 50% of the GI cancer, actually present a huge commercial potentials. So what we find, or our finding is we find Cadherin-17 or CA17 is a very attractive cancer target because it's highly expressed in the tumor but not in the normal tissues. There's a plethora of journal and article published not only from my lab and also outside our lab has been shown Cadherin-17 is a very promising target for GI cancer, and actually that was reviewed in the Nature Cancer Review article years ago. And Cadherin-17 is a superior target because when you look at this profile, Cadherin-17 is expressed over 95% in all GI colorectal cancer. In fact, in our phase 1 clinical trial, we find all the colorectal cancer has been shown positive for CA17. And versus the other like gastric cancer, pancreatic cancer, also about 50 to 60% positive. Compared to like the Claudin 18.2, I think we talk about these drugs and also the other markers. So we strongly believe, and we have from our experience, Cadherin-17 will be a superior target for treating GI cancers. And our company focus on 2 areas. One is generating cancer immunotherapeutics. And on the other hand, I think we need to provide and offer a personalized healthcare. So at the same time, we're also providing a kind of a screening test and also a companion diagnostic to guide the physician whether this patient are selected or suitable for being treated. So our company has developed 3 technology platform. One is a multi-specific platform, and the second one is an advanced cell therapeutic platform. And the third is the diagnostic personalized healthcare platform. So as a biotech company, it's very important. The patent is our asset. So we have submitted 10 or published 10 PCT, and we have a number of granted and also under the pending approval. So our patent covers not only the target and also the therapeutic molecules, the diagnostic, and also the platform technologies. So back then, I think we are very fortunate. We are able to file patent on the target. Nowadays, almost impossible can file any targeted patent. So this is our pipeline showing our current leading asset is ARV-202. And we also get a drug link from WHO already, but I think we're going to announce later this year. So we are already in phase 1A. I think the data have been very promising. So we are going to advance to phase 2 next year. And so this is a clinical trial roadmap. So as I mentioned, we started we dosed the patient in Australia last year, and now we just got the DMC approval. The clinical trial have been saved, and we can advance further to the next stage. So I think when we were very down, when we are running out of resources and money, sometimes we receive a letter from the patients. So this patient really gave us a very momentum to keep doing and keep working. So some work didn't say, actually a patient admired me like an idol, one of her idol is removed from here, but I think it's very important for us to really discover and make a new drugs to treat the patient. This patient actually is a Swedish family, went to Australia to receive the trial, so she has stage 4 gastric cancers. So, and also, I think commercially, I think we need to position where our drug will be once approved. So this is from the data monitor health modeling, showing that if given ARV-202 approved, so our drug will be very promising. So this is the highlight about our company. So if you heard from the ESMO, so yesterday, actually the DLL3, CD3 bispecific T-cell engagers have made a very big news because they show that about 40% ORR in phase 2 with very low dose 10 mg in the patients. So I think time's running out. I'd like to share when I was a faculty in the university, so I usually stand in the front, in the center. But now as a company CEO, I try to stand at the back to push, to propel the young generation to be the frontiers. So thank you so much.
Thank you, Dr. Luck. Now we'll welcome our final speaker before we turn it over for discussion. Dr. Andrea Vanelsis is the CSO of Manatee Bio.
Short, so we have a lot of discussion. So, Andrea Vanelsis, I'm the interim CSO for this company. It was founded about 3 years ago, based on technology that came out of Hopkins, and I'll refer to that later. I'm also a partner at Third Rock Ventures. We built about 2.5 years on this company, and it's just did its first Series A close for a significant amount of money. I'll come back to that. So essentially, since the introduction of checkpoint inhibitors into clinical development, we're starting to segment solid tumors, at least, into 2 categories. You have the hot tumors and the cold tumors, and hot tumors clearly respond to checkpoint inhibitors, at least to a certain extent. Cold tumors do not. And these, the category of cold tumors consists of large indications that currently do not receive any benefit from immunotherapy by and large. But these tumors are cold not because there are no T cells. In fact, if you look carefully, there are T cells. And these T cells simply do not see tumor antigens or not enough tumor antigens to go after. So what if you could redirect all these T cells? These T cells are influenza specific, they see COVID, they see EBV, etc. They're ready to go, they're memory cells and not exhausted. And this obviously is the realm of T-cell engagers. Now, T-cell engagers have been tried and tested for a long time in solid tumors, have not really met their potential yet.
And the reason we believe is because they've not gone after truly tumor-specific targets. So there have been some really encouraging data. Actually, you just reflected on this a while ago. DLL3 and a few other targets have shown clinical promise. But however, most of these trials suffer from cytokine release syndrome, usually related to on-target off-tumor toxicity. So the founders of Manatee started working on this a long time ago, and you'll recognize the names. Drew Pardol's in the room here, Bert Vogelstein and their colleagues started working on this probably about a decade ago, this idea of are we able to specifically target tumor cells by making use of oncogenic driver mutations as presented in HLA class 1. That's a tall order. These are low-density targets. But we knew these are immunogenic and antigenic because patients actually have been known to contain, that their tumors have been known to contain T cells that see these mutations. So we know that these mutations are there, they're productively presented, and can be effectively targeted. So how do we do this? And so what are some of the numbers associated with that? So microsatellite stable colorectal cancer has been mentioned a couple of times. Steve Rosenberg referred to some of these mutations. These are actually the shared mutated neoantigens that can be found in these tumors that we could develop drugs to if we do it right. Importantly, these are also oncogenic drivers, so it's difficult, maybe not impossible, it's very difficult for the tumor to get out of this by shutting down protein expression. And they're found frequently in a variety of cancers. So our first targets are mentioned here. The p53R175H mutation is a fairly common mutation in p53, found in 6.5% to 7% of microsatellite stable colorectal cancer patients, newly diagnosed. On an annual basis, we find a couple of thousand of patients. If you take this beyond microsatellite stable colorectal cancer, which is the promise of these drugs, you could look for tumor-agnostic approvals. You're thinking about 10,000 patients, by and large, newly diagnosed in the US only. And that doesn't take into account prevalence or ex-US patient populations. So how do we do this? We use a platform or a protein actually that was developed by our founders. It's initially when they came to us about 2.5 years ago, they had very cool killing data, cytotoxicity data in vitro and in vivo, specific, highly specific to the mutated peptides as presented in HLA class 1. And these were, the cytotoxicity was induced by a protein called, that was referred to as a single chain diabody, a very small, rigid protein structure that is very well capable of detecting low density targets. You should think about less than 100 targets per cell, maybe even less than 10 targets per cell. T cells can do this, and the only other modality that I know of that can do this was the single chain diabody that was developed by the Hopkins founders. I was not very comfortable developing a single chain diabody, so we set out to seed this company together with the other founding investment firm called Catalio Capital. Together we seeded the company and started to build on this basic structure. Can we build this into something that looks more like a drug? We ended up with an asymmetric antibody structure that maintains all the specificity we had. It was absolutely specific only for that mutation. It did not see wild-type p53, for instance, as presented in the same HLA class 1 molecule. It also maintained the same sensitivity. We can still kill cells that contain less than 10 targets per cell. And we now have the added features that we can dose these once every 2 or 3 weeks. So we turned this into a half-life extended molecule that looks like an asymmetric IgG. It has all the features and capabilities and capacity that an antibody would have. It produces like an antibody. It purifies and is stable like an antibody, but it has this very peculiar low-density, ultra-specific targeting capability to redirect all T cells we find in a tumor to these oncogenic driving mutations as presented in HLA class 1. Our first molecule is called MNT1025. It targets this p53 mutation in HLA 2.1. Also sees, by the way, this mutation in other HLA-A2 suballeles. And it engages all available T cells to start killing this tumor. And the added feature to this principle is now we can select our responders based on molecular genetics. This is actually the molecular genetics a lot of our patients are now being subjected to when they're first diagnosed. That allows us to do very targeted clinical studies. I mentioned this, the only other way to target these mutations probably is by using T cells. But that requires a lot of extensive culturing and technology, and we've sort of dumbed it down to an off-the-shelf protein therapeutic that we now know we can exchange. The blue domains shown in the structure are actually the peptide MHC targeting domains. If we take out the p53 targeting domain, put in a KRAS G12V targeting domain that sees this mutation in HLA-A3, we end up with something that looks like a drug. So it's a nanomolar potency, long half-life antibody drug that, so what we think this platform is actually a really nice plug-and-play platform that we can extend. So we've just closed our first Series A. We don't need more money, but we need really a lot of talent to build out this company the right way. We need the right scientists and translational immunology and translational medicine people. So if you know of somebody who's interested in joining the company in Boston, let me know.
Great. Thank you, Andrea. So you finished? So we have a few minutes for discussion. Love to get some audience questions. I'm going to open it up with a question that I have for each of the panelists. We didn't plan this, so see how you guys react. Although, Andrea, you just answered the question already for yourself. We have a room full of incredible thought leaders and a community of fellow entrepreneurs. Many of us are staying for another 6 hours, so you have a captive audience. Under 1 minute each, can you tell us a current challenge that your company is facing that you think someone in the room could help you with? And then I'd encourage everyone, come up and find these folks after in the next few hours and help them. One rule, only one person at most can talk about needing funding. You already said you don't need funding. Who wants to go first?
Funding.
Okay, Dr. Johnson, you've elected to go first.
I don't need to go first. I was joking.
I can go. For us at .bio, as I mentioned, we are based in Singapore. Obviously, Singapore is a very small market. We're aiming to reach elsewhere globally, and obviously the US would be great. We're looking for people that can help us accelerate that through connections, through expertise, through access to clinical development or clinical ideas. So I think there is a lot of people here in this room that could help us in many ways. So we hope that some of you would come and help us achieve the potential for the company. Thank you.
And your model is fundamentally based on partnering, as far as I understand.
We do. So we accelerate a lot of development through partnering, but eventually we want to also push our internal pipeline. Obviously funding comes to play, but also the potential for the technology is quite broad. So we want to be able to de-risk some of the molecules through partnering, and then that gives us the confidence to push some internally as well. Yeah.
That's great. Stacy, thoughts?
So I will sort of roundabout get to funding, which is, we're drugging a first-in-class target in probably the most well-validated pathway in oncology, but we still get a lot of pushback. It's never been drugged before, so I want to see clinical data. So I guess if I can't ask for funding, I would say I want other leaders in the field who recognize why we need to drug CDK2/4/6 simultaneously, which I think we all agree on, and recognize that it's been difficult to drug it with small molecule ATP inhibitors, although people keep trying to do that, and we probably will end up with the same results that we've had for the last 40 years. So if you acknowledge those two things, then you've got to be innovative, and drugging P27 is the innovative way to do it. So KOLs that will continue to support this, to talk to the VC funds and say, yeah, this is the way to finally crack this resistant problem. So that's
great. We've got all night to help assemble your next panel.
Yeah, there we go.
Jeremiah, thoughts?
All right. Well, I think from the perspective of what we have as a lead candidate, we're pretty well set, at least to get to the IND ready stage. We're going to need some money at some point to get to the clinic and the manufacturing, but I'm not supposed to talk about that. But what we're really poised to do right now, I think, is to show off the versatility of our platform. And even just today, I wrote down probably half a dozen ideas based on the talks that I saw, and I will come find you, where I think our platform, when coupled with either a payload you're developing or an antibody that you're working on, I think could really offer synergistic benefits. And so right now, I think that's what we'd like to do, is work with others to just show how many iterations of this platform we can develop. And I say this, it sounds maybe, I don't know, if this doesn't work out this way, I apologize in advance, but if you come to me and you have an antibody that you would like to develop it into an ADC or a new drug, I would say we could get you a construct to test in about 2 weeks or so. So, it's fairly streamlined now, the manufacturing of this platform.
That's great. Oh, that's excellent. John, as another one of our international partners?
Yes. First of all, I think I thank Suzhang and AFCL give us the funding to breed us to be successful, and now we need a harvest. So I think the problem I need now is a lawyer and CFO. Okay, lawyer, because we have a deal, we are going to negotiate with Big Pharma. We are always become disadvantageous because we don't have the expertise to negotiate with a Big Pharma. And also we need a CFO, won't charge us high tickets, but I can guarantee he or she will get stock options when we are IPO in 2 years.
John, you're going to be very popular at the dinner tonight. Andrea, do you want to, you spoke to, I think, a very sincere need that we all feel in this region and everywhere for talent. Do you want to put a finer point on that in terms of, you guys are building pretty fast? And
Yeah, I think maybe I'd like to expand it beyond Manatee, because I'm in venture, I build a lot of companies. But I go back to my own experience. I was actually trained and mentored by the best of the best, in hindsight. I didn't know that at the time, but I was. I worked with Jim for about 18 months in Berkeley while he was doing all this wonderful work. I didn't realize how important it was, but it did inspire me to go into pharma and to learn how to develop drugs. I learned everything I know today from working in pharma. And then I decided, pharma is good for some things, but not for other things. I decided if you want to do early discovery and translation, it would be better done in biotech. So then you have to work with people that tell you, that explain to you how you build a company, how you raise money, how you do it wisely, how you keep focused, et cetera, et cetera. So I learned a ton from a lot of different people. Drew was in the room, is in the room. I worked with him for 25 years in different occasions. I think it's really important that all these great minds here train people and train them the right way. It's very important because I can tell you, I meet so many people that go out and have a bit of training, but not enough. And biotech is not a great place to learn everything. I think we should all recognize that pharma has a purpose, and pharma is a great place to learn how to best develop drugs and put them on the market. But we're scientists, so we like to be in the beginning. So there's a purpose for pharma, there's a purpose for biotech, there's a purpose for academia. But please train your people. Please train them.
beautifully said, very, very nicely said. Do we have, and I apologize, each one of these talks really could justify 5 or 10 minutes of deep technical questions, so we don't have time for that today, but do we have some questions from the audience? I can't see that well. We don't. Dimitra. I can recognize your voice, I can't see you, Dimitra.
Yeah, I have a question maybe for all of you. As you guys know, we're going through what is considered like a winter for VC investments. So what advice do you guys have for sustaining and like managing through this situation?
Andrea.
So, first of all, this is the best time to build great companies. It may be hard to get money, but it forces you to really think about, what am I trying to do? What am I trying to establish? How do I explain to other people that it's important that they put their money in? It's also the best time to find great talent, because a lot of companies are folding. They were folding for many reasons, sometimes not because they weren't started the right way, or some of the drugs don't work out. We know that most of our drugs don't actually deliver what we expect them to deliver. So I think this is the best time to build a company. But it also forces you to be very specific as to why you're building it and what you're actually building, who's benefiting from it, what patient is waiting for my drug, what patient is waiting for this solution. And I think in times of plenty, I think we're building and financing a lot of companies, and you'll look back and see then not many of these companies actually turn out to be successes. Even from a financial perspective, they're not always successes for the original investors. The companies you build in times of frugality and when it's difficult to find money, those tend to have a slightly better chance, in my opinion. And I think I'm telling you this after 7 months of talking until I was blue in the face to try to raise my 130 million, which is crazy. This would never have happened 3 years ago. We would have done it in 2 months. But that's the reality. You have to have a really good story, and you have to think about, what am I going to do with an investment? Who is being served, and is that actually a worthy cause? And then you can find money, because there's so many people that made so much money in biotech and life sciences over the past 2 decades. They're still intrigued and eager to invest. Maybe not automatically, but they need some convincing.
Wonderful. Maybe we'll end on that and thank our panelists once again for their great talks.